Welcome friends, I am Dr. S. Megala, Assistant Professor, Department of Microbiology, St.Joseph College of Arts and Science, Autonomous, Cuddalore. Now I am going to talk about pure culture and preservation of microbial culture. First one, preparation of medium. Take a clean conical flask containing distilled water as desired quantity for media preparation. Add the necessary media ingredients into the distilled water taken and mix it well by keeping in the rotatory shaker. Next, check the pH of the medium. Put the non-absorption cotton plate and wrap with aluminium foil. Next, sterilize the medium by keeping the flask into the autoclave for 121 degrees Celsius for 15 minutes at 15 lbs pressure. The sterilized media can be aseptically dispensed into sterilized container. Let it allow solidifying in case of solid media, and now the media is ready for microbial inoculation. Next one, pure culture. A culture is defined as the active growth or cultivation of microorganisms in nutrient media. But the pure culture is defined as the population of only one species of microorganism, all derived from a single parent microorganism. The isolation of one kind of microorganism from a mixture of many different kinds is called the pure culture technique. Only with the pure culture, properties like cultural, morphological and biochemical properties of individual types of microorganisms can be examined. A number of techniques have been employed for the isolation of microorganisms from natural environment. They are: first one, serial dilution; second one, stick plate method; third one, pour plate method; fourth one, enrichment culture. First one, serial dilution, about 1 ml or 1 gram. Liquid means water, solid means it's soil. Of sample is transferred to a test tube containing 9 ml of distilled water and mixed well to get 10 to the power of 1 dilution. 1 ml of homogenized sample from 10 to the power of 1 dilution is pipetted out into a test tube containing 9 ml of dilutants using a sterile pipette to get 10 to the power of 2 dilution. The procedure is continued using sterile pipettes for each dilution up to 10 to the power of 7 dilution. Next one, pour plate method. 1 ml from 10 to the power of 4 dilution for fungi, 10 to the power of 4 and 10 to the power of 5 for actinomyces, 10 to the power of 5 and 10 to the power of 6 for bacteria was pipetted out and added into the petri dishes containing appropriate medium. This whole process should be done under strict aseptic condition. After addition of 1 ml of the plate should be kept on the incubator at the room temperature. After incubation, the bacterial or fungal colonies are counted and recorded. Third one, spread plate method. It is an isolation technique. It is a modification of pour plate technique. In this method, the mixed culture is serially diluted in sterile distilled water. A small amount of mixture is then poured on the surface of an agar plate and it is spread evenly using a sterile glass electrode. The isolated cells grow into colonies. Fourth one, stick plate method. Stick plate are prepared by sticking a small amount of mixed culture over the surface of the solid medium in a petri dish with a platinum or nichrome wire loop. Successive sticks thin out the culture sufficiently. After sticking, the plates are incubated overnight so that the transferred organism grow and multiply and give visible colonies. These colonies originates from single cells, thus creating a pure culture. Fifth one, enrichment culture. This method is often used if the microorganism to be isolated is present in relatively small numbers or its growth is slow. This will favor the growth of particular type of microorganism but will be unsuitable for the growth of other types. This technique is particularly very useful in isolating organisms which can degrade a variety of pollutants that accumulated in the environment, especially in water and soil. Sixth one, selective medium. It is an isolation technique, a selective medium containing a chemical which suppress or kill unwanted species, but the medium allow the desired organism to grow. For example, crystal violet inhibit gram-positive bacteria. When crystal violet is added to the medium will select the gram-negative bacteria. Seventh one, differential medium culture. The differential medium gives different appearance to different species. For example, in EMP agar medium, E. coli will produce green metallic sheen colonies, while Aerobacter aerogens will produce pink colonies with dark centers. Next one, single cell isolation. In this method, individual cell is picked out using micromanipulator and microscope. Next, we are discuss about preservation of microbial culture. When you have developed good strains of microorganism, it must be preserved properly so that it could be used in future also. If the culture have not properly been preserved, their characteristics future may decline after certain period. Therefore, several methods have been developed for maintaining in the organism in viable condition over a long period of time. These methods vary according to strains. First one, agar slant culture. Agar strap or slants are prepared after autoclaving the medium. They are inoculated carefully with individual culture and incubated overnight. Then culture are stored at 5 to minus 20 degrees Celsius. It must be subcultured periodically once in a month. Next, period of transfer vary with species. Disadvantages: mutant may be developed upon exposed radiation. Mutants means the sudden inheritable changes, that is called as the mutant. Next one, agarstlan culture covered with mineral oil. The bacterial culture can be stored alive for 2 years. Some can be 15 to 20 years in the mineral oil. The culture kept in agarstland. The sterilized mineral oil is poured into the agarstland. The oil level must be above the tip of the slant surface. The oil covered slant is stored at 5 degrees Celsius. The role of oil are to prevent cells from dehydration and slow down the metabolic activities. Third one, storage in saline suspension. Bacteria require salt for various metabolic activity, but at higher concentration, salt acts as the bacteriostatic. Hence, bacterial culture is preserved in 1% salt concentration in screw-capped tubes to prevent evaporations. The tubes are stored at room temperature. Fourth one, preservation by drying in vacuum. The culture are dried over calcium chloride in a vacuum, then stored in a refrigerator. At such conditions, the organism survive for long period than the air-dried culture. Fifth one, cryopreservation. Cryopreservation means the cultures are grown in culture tubes. Then 10 to 30% of cryoprotective agent, that means glycerol or dimethyl sulfoxide, is added into it to protect the culture from cold shock. The culture is dispensed into small vial. The vial are properly sealed and kept in liquid nitrogen at minus 176 degrees Celsius to minus 196 degrees Celsius. Next one, lyophilizations or freeze drying. In this method, the microbial suspension is put in small vial and frozen through drying under vacuum. Vacuum drying result in sublimations of cell water. Then the vials are sealed off in vacuum. The lyophilized culture are stored at 4 degrees Celsius. The culture remain viable for about 30 years. By this method, a large number of culture are maintained without variation in the characteristics. The advantages of the lyophilizations. First one, less space enough for storing lyophilized culture. Can be sent through mail. Reviving ability is good. Disadvantages of the lyophilizations: instrument used are expensive. Next one, maintenance in formaldehyde. Agar plate culture can be preserved by placing a drop of formaldehyde on the inner side of lid. It is stored in a refrigerator at 4 degrees Celsius. Next one, storage in sterile soil. Soil is sterilized and bacteria are mixed with the sterilized soil. The mixture is dried at room temperature and stored in the refrigerator. Spore forming bacteria and fungi can be stored for about 80 years by using this method. Next one, storage in silica gel. Silica gel is powder sterilized and cooled. Bacterial cells are mixed with silica gel powder and stored at low temperature. Bacteria and yeast can be stored for about 2 years in this method. Thank you.